Okay. Hello, everyone. Welcome to the first INC talk of the summer. I'm Mike. It's Shyam. Shyam's been here for a little bit. He worked with Chuck Stevens for a while, and now he is working on a rat atlas project and figuring out what else is happening. And without further ado, here's Shyam's talk.
Okay. Thanks everyone for coming from near and far. Where to start? I'd like to say that this project that I'm going to present is a collaboration with Saket, who's at Cold Spring, Alex Fleischman, and Simon Dust, who's at Brown. They're the mice experimentalists, and also fly experimentalists, Len Turner and Merhab at Geneva. And in this project, we got interested in understanding how you can do perceptual learning or fine discrimination. There are lots of terms for this, but the basic idea is that you have two stimuli that are nearly identical, and initially they're indistinguishable, but with lots of practice experience, you are able to tell them apart. And this is something that is a phenomenon is common to our everyday lives. For example, if you drink wine, me, for instance, I can tell a red from a white wine because the color is different. But if you give me two red wines, I probably can tell them apart. But there are people, maybe some amongst you, who have a lot of experience with wine, thousands of hours of practice, wine connoisseurs, and you can tell the difference between like Merlot 2000 and 2010. So how does it happen? So one way this could happen is if you look at, you have a neural representation of these stimuli in some region of the brain, and that's how they look. And because of that, there's a high degree of overlap. So it's tough to tell one stimulus from another. But with learning, you're able to tell this overlap reduces. And since the overlap is smaller now, you're able to tell them apart. So in this talk, I'd like to give you our proposal or our idea that this learning is using, there is noise or variability in responses, and it's using this variability to kind of pull the two, or in this case, orders, but in more generally, stimuli apart. And to tackle this question, we decided to use the olfactory system. So there are three reasons we use the olfactory system. First is that what I just described, the phenomenon of fine discrimination, is being observed with olfaction. So this is an experiment that was done in Don Wilson's lab. What they did was they took 10 mixture odorant, and they generated two new mixtures. The first mixture, they just took out one of the 10 odorants, and the other one, they replaced one of the odorants. And then they trained the mouse on a go or no-go task, or sort of on the right, you see they trained the mice so that if it gets the original mixture, it goes left. If it gets the changed mixture, let's say the 9-component mixture, it goes right. And here's the result. So on the x-axis, you see the days post-training. The leftmost point is vanilla mint. It's kind of used as a control. So that's the mean error rate. And on the y-axis, you have the mean error rate in choosing. So that's the control is the mean error rate that you should be aiming for. So as you can see, in trying to distinguish the 9 and 10 odorant mixture, the mice has problems on the first day, on the second day, and I'm not showing this to you, but in subsequent experiment, they showed that it takes up to a week of training for it to be able to distinguish the two mixtures. So it's initially unable to distinguish the two odors, but with a week of practice, it's able to distinguish that, which is fine discrimination. The second reason we chose the olfactory system is that the pathway from the nose to the piriform cortex is well known, and it's all the connections and the responses are mapped out. So it's easy to model and look at input and output and see the relations between them. And importantly, Wilson lab also showed that the piriform cortex was implicated in fine discrimination. So let me just give you an idea of the olfactory circuit. So the first stage, you have the olfactory epithelium in the nose, and the olfactory epithelium, you have different kinds of olfactory sensory neuron types that sense different components of the odor. So mice have about 1,000 olfactory sensory neuron types, and each one is particularly tuned to a class of chemicals, let's say alcohols or ketones. It sends its information then to these circular neuropil in the olfactory bulb called glomeruli, and these then send to receptor neurons called microcells, send information to the piriform cortex in the brain. So the third reason we chose the olfactory system is that the structure I just described, or the architecture I just described, is highly conserved, and it's also observed in flies. So here in the fly, the antenna and the maxillary palp function as a nose, and there are about 50 olfactory sensory neuron types. They all send their information to these blue circular structures or neuropil in the antennal lobe called, again, these are glomeruli, the fly glomeruli. And there are 50 of them, which match the 50 olfactory sensory neuron types. And they send their connections to various structures, but most importantly, they send their connections to the mushroom body, which contains Kenyan cells that encode experience-dependent odors. And the important thing with the mushroom body is also that it does learning and discrimination. And since the fly system is more tractable, the structure that does learning and discrimination has been really well mapped out. So it allows us to ask questions about how fine discrimination can take place, because we can manipulate learning and discrimination. Okay, so that's the olfactory system. One of the features of the olfactory system is that the representation of odors in all three stages uses a combinatorial code. So here, let's say you have a specific odor, and here's the response to the odors in the oceans of the glomeruli on the left. It looks like this, and you have a particular combination that responds to this odor. If you take a different odor, there's a different combination that would respond to that odor. So the combination that responds to different odors is unique. And this combinatorial code is then passed on to the next stage, the mushroom body, or the piriform cortex, and it's encoded by Kenyan cells or piriform cortex cells. And that's also a combinatorial code. And in this stage, in the fly, this combinatorial code gets filtered through a winner-take-all circuit. So there is a giant inhibitory neuron called anterior paired lateral or APL neuron. And the way it works is that all the cells, all Kenyan cells, send excitatory input into the APL neuron, which then negatively feeds back and suppresses all but the highest firing neurons. So it kind of sparsifies the code. These Kenyan cells, the sparse code then is sent to various mushroom body output neurons. They all fall into two types, so I'm just abstracting it to these two neurons, approach and void. And the synapses between the Kenyan cells and the approach and void neurons are subject to plasticity. So that's where learning and discrimination takes place. And whether the animal approaches or avoids an odor depends on which neurons are firing at a higher rate. So it compares the two neurons and then it approaches and avoids. So having given you an introduction to the olfactory system, I'd like to give you a brief idea of what I'm going to cover, the three topics I'm going to cover in this talk. First, I said there is noise or stochasticity olfactory system, and I'd like to show you our characterization of what the stochasticity looks like. The second part, I'll talk about how this could be used by learning to improve discrimination. The third part is looking at how the circuit can generate the stochasticity or the stochastic code that we observe. Again, these are all my collaborators, and I'm also happy to say that our paper just got accepted, so maybe if you're interested, you can read more about it soon. All right, so coming to the first experiment. So first experiment, which kind of inspired the whole project, was an experiment done in Glenn Turner's lab when he was at Cold Spring Harbor. The first author is Rob Campbell. And in that, what they did, one of the things they did in the paper was they recorded responses from the fly mushroom body, the Kenyan cells. And they looked at catching responses. So they used GCAM. And they exposed the animal to an odor and they did several trials or exposures to the same odor. And what they noted was, so this is the first trial to some particular odor, and they did subsequent trials, and they noticed that the neurons kind of, if they responded in one trial, the subsequent trials, some of them didn't respond, and they responded again. So there's some variability in this population response over several repeated exposures. So they broke down these responses into two types of cells. So the first type of cells, they didn't call them reliable, but we call them reliable cells. So these reliable cells are cells that respond in more than half the trials. So here are two examples of reliable cells. The first one and the second one, they respond in 3 out of 4 trials. The second set of trials are unreliable cells that respond in less than half the trials or half the trials. And here are 2 examples where you see cells responding in 1 out of 4 trials. They shared their data with us, and we were able to then analyze the data a little further. And here's a plot of the data. But first, what is the data set? So they recorded responses in the fly to 6 odors and 6 trials per odor, and they recorded from 124 Kenyan cells. So we took these cells, and on the x-axis, you have reliable cells and unreliable cells. On the y-axis, you have percentage of the population that's reliable and unreliable. As I said, 6 orders, 6 trials, and this is a plot per trial, so there are 36 grade points in here. As you can see, there are more unreliable cells than reliable cells. So the number of reliable cells is about 5%, and the number of unreliable cells is about 8%. As I said, the olfactory structure is conserved across animals. So we also looked at responses within the piriform cortex in mice. And this was data generated by Alex Fleischman and Simon Dest. And over here, again, we looked at the cells, and we analyzed them. And here's the graph of, in this case, we have 8 odors, 8 trials, 288 cells. And again, on the x-axis, you have reliable cells and unreliable cells. On the y-axis, you have a percentage of cells each trial that's reliable or unreliable. And as you can see, there are more unreliable cells than reliable. Over here, the number of reliable cells is about 3.5%. The number of unreliable cells is about 9%.
You mean in each odor, not in each trial, because
Sorry, go ahead.
because you realize if they're reliable throughout the trial, not one.
It's a good question. I'll show you another graph, but this is per trial. So what we did was you take the 8 trials, and then you find out all the cells that were reliable and all the cells that were unreliable. So reliability and unreliability is a property per order, as you said. And then in each trial, you can see the number of cells that were from the unreliable set and the reliable set. That's what I plotted. We've also, I don't have it here, but we also have a plot per order, and it's the same relation. It's actually, yes.
So in the cartoon, there were 2 reliable cells, and in the cartoon, they both didn't fire on the same trial. So are there correlations in when the reliable cells are not active? Like are there across the population correlation?
Good question. As far as we could see, there were no correlations across the population. I don't have the data to show you, but we looked at it and some, you could be reliable for one odor and unreliable for another odor, or just silent for another odor.
But for the same odor,
For the same odor, there are correlations. Okay, yes. Actually, I have a graph showing there. So, good question. All right. So this is kind of getting at your point. So I talked about reliable and unreliable cells, and the distinction is a little bit arbitrary. It's based on tradition because that's how people classify cells that they reported. But the property the reliability property actually is more graded property. And on the graph here, we can show that. So on the x-axis, you have reliability. On the y-axis, you have the number of cells in each reliability class for the whole set. And here, reliability is calculated on a per order basis. So you can see that the less reliable.
Sorry, Shyam, to interrupt. Can you explain one more time? You went a little bit fast for me. What do you mean by unreliable cells? And why do we have more unreliable than reliable?
Okay. All right. Unreliable cell. Okay. Reliable cells are those. Okay. I gave you a definition of unreliable versus reliable, but let me give you a fresh definition. So you have 6 in the fly, you have 6 trials per order. And let's say you have a cell and it responds in 2 out of the 6 trials. So its reliability is 1/3. And you have another, let's say you have a second cell which responds in 5 out of the 6 trials. So its reliability is 5 out of 6. So in the graph here, on the x-axis, you see reliability going from 1 to 6 because you have 6 trials. Does that make sense? Sarah?
Yes.
Okay, perfect. Now, if you have more questions, just please feel free to ask me.
And why? And then my second question was why
Are there more unreliable?
more?
Why are there more of the less reliable cells? That's your
Right.
question?
Exactly.
Okay. Let me go further through the talk. If the talk doesn't answer your question, we'll come back to it at the end. Because I have an answer for that a few slides later. Okay.
Sounds good.
Okay. All right. So on the fly, you can see that reliability is a graded property. As reliability increases, the number of cells in each reliability class decreases. And we find the same thing. We also did an analysis of the mouse data set, and you can see as reliability increases, the number of cells in each reliability class reduces. We also looked at response rate.
So can you go back?
Sure. Yeah.
Well, that's really weird. So there's a little uptick on both at 6.
On between 5 and 6.
So like 6 is higher than 5 and 6 is higher than 5 in the mouse as well, just by.
Okay, 6 is not it's about the same level as in the mouse. So 5 being higher than 6, it could be that some of these cells responded like maybe the GCAN signaling was off. They responded in more like all the trials. There were some in the fly data set, like 1 or 2 cells. That's why you see that.
I see.
In the mouse data set, they're at the same level. I wouldn't.
I wouldn't comment.
There's no
It looked like you can get 5 or 6 on both.
Okay. So yeah.
One more follow-up question. So
Sure.
cells that don't respond to any odors,
Mhm.
are they considered highly reliable?
Of course, yeah. Well, they do respond the same way, but then they're not. Okay, there are 3 classes of cells then: reliable, unreliable, and silent cells. So
So you're not looking at the.
yeah, I'm not considering them as cells.
Okay.
Okay, so then we also looked at response rates of cells in each of the reliability classes. And we calculated response rates as the average response over the 6 trials. So on the x-axis, you have reliability each of the reliability classes 1 to 6, and on the y-axis, you have the average response of all the cells in that reliability class. And as you can see, as reliability increases, the response rate also goes up. And this is also true in mice. As reliability increases, the response rate goes up. So to summarize what I've just shown you, we have about from each data set, both mice and flies, we have about 4 to 6% of cells that are reliable. About 30 to 40% of cells are unreliable, and you have a lot more unreliable cells, but the reliable cells have a higher response rate. Okay, so given these two classes, one of the questions we had was, could they be coding different kinds of information, or could their responses be different? So to test that, we had in the fly, we had a data set from Hallen and Carlson where they had recorded responses in olfactory sensory neurons in the fly nose to about 110 odors. And those odors, the odors that we looked at were part of their data set. So we could compare the similarity between odor pairs in the nose and see if the similarity was reproduced in the mushroom body. And in order to look at, when we looked at the mushroom body cells, we broke the cells into reliable cells, a reliable cell set, and an unreliable cell set. So here's our result for the reliable cell set. So on the x-axis, you have the nose OSN similarity score. On the y-axis, you have the reliability similarity score for reliable cells. And each circle is one odor pair. So there, and actually in this case, we had about 8 or 9 odors. So there are about, I think there are about 30 to 40 points there. And as you can see, when the similarity between odor pairs is low, I should also say this, we calculated similarity by just taking correlation, Pearson correlation coefficient of the two vectors in the nose and the mushroom body. So when the similarity is high in the nose, it is also high, or let me go the other way. When there is no similarity in the nose, odors in the mushroom body responses also have very little similarity. They're decorrelated. And as the similarity in the nose increases, the similarity as represented by reliable cells also increases. We then looked at unreliable cells, and over there you can see that unreliable cells are decorrelated both for similar odors as well as dissimilar odors. So the thing is, we had this Hallen-Carlson data set for flies, but we didn't have a corresponding data set for mice. So we came up with a second measure, a different measure. What we did was, as a matter of control, we first looked at the fly data set, and we took all odors that had a correlation greater than 0.5. We classified those as similar odor pairs. And then we looked at the reliable and unreliable cells. So on the x-axis, you have unreliable and reliable cells. And on the y-axis, you have the correlation of the Kenyan cells for these similar odor pairs. And you can see the unreliable cells are highly decorrelated, but the reliable cells are correlated. This is an analysis we carry out in the mice data set too. And over there, you see a similar property. Unreliable cells on the x-axis, you have unreliable and reliable. On the y-axis, you have similarity measured by correlation. And again, here you can see that unreliable cells are decorrelated a lot more than reliable cells. We came up with a second measure to calculate, to test how similar or different the reliable versus unreliable cells are. So let's say you have two orders A and B, and these are your responses to the two orders. We calculate overlap by taking responses of, let's say, cell X, order A, and order B. And let's further say that the cell responds 4 out of 6 trials for A and 2 out of 6 trials for B. So the probability of response would be 2 by 3 for the first one, 1 by 3 for the second one.
What are the colors?
Oh, sorry. The colors, the box colors and the grid box colors represent the intensity of fire. This is the schematic, so it's made of color. But in general, that's just to give you an idea, because the first one is reliable. And as I already showed you, reliable cells have a higher response rate than unreliable cells. Okay, and then we calculate the overlap measure as a probability that the cell will respond to both of them, and that's 2 by 9. And then we can sum up these responses for the whole population. There's one cell, but we can sum up all 124 cells. And here's the, oh, and the other thing we did was we calculated the overlap score that way. And then we did a second calculation where we randomly shuffled the cells and again calculated the overlap. So it's, and then we normalized the score to the random score. Does that make sense?
You get to say it again.
Okay, sorry. We did, let me go back. So we did 2 measures. We calculated the overlap using actual responses. So that's the overlap score. And then we also calculated the overlap score for the whole population if the cells had been chosen randomly. And so we normalized the overlap score for actual response by no overlap score for random population. So it gives you a measure of how far it is different from if the cells were just chosen randomly. And you can see that if you look at each of the reliability classes on the x-axis and you look at the overlap, the normalized overlap score on the y-axis, as the cells get more reliable, the overlap score increases. So you can look at it in 2 ways. More similar odors, the cells are more likely to respond the same way, and whether the odors are similar or dissimilar, the unreliables, if your cell is less reliable, it's more likely to be decorrelated or respond differently. And we did the same calculation for mice, and that's result. It gives a similar result as reliability increases on the x-axis, overlap also increases on the y-axis. So to sum up, what we have are, if you look at responses over a period, over a number of trials, you have a bunch of cells that go from less reliable to most reliable. And the most reliable cells have the highest response rate, but they're also the smallest class of cells. And so coming to the second part of the talk, our hypothesis, I'd like to present our hypothesis for learning. It's that these unreliable cells are more likely to be decorrelated between similar odors. And they're also more, even though they have lower responses, they're more numerous. And learning uses these cells to decrease the overlap between similar odors. So if I can give you an idea, so you have 2 odors, A and B, and you take a Venn diagram of that overlap. If you just consider reliable cells, there's a small amount of overlap shown in yellow, and it's easy to tell these 2 odors apart just using reliable cells. But if you take similar odors, there's a high amount of overlap, and it's hard to tell them apart. But now, if you include unreliable cells, you increase the size of your odor set. So, and you also increase the amount of, or you decrease the amount of percentage of overlap. Well, even though here the absolute overlap area is the same, percentage overlap area is smaller, and you can distinguish the 2 odors. And we'd like to, our hypothesis is that this learning actually recruits these unreliable cells. So, yes.
So does this assume that the unreliable cells responding to similar odors, their variability is not correlated?
Yes. I mean, it's not an assumption, but I don't have it here, but we did look at their variability. and it doesn't seem to be correlated. I mean, the variability is not correlated. The variability doesn't match the reliability class. Does that make sense? So if you plot the correlation between cells and you look at what do you call it, cross reliability classes, there is correlation, but it's about the same for all the classes.
So it's the same for all the classes. That means that this. Wouldn't you expect when you include also the unreliable cell, this diagram just to be bigger but the same percent overlap? If the responses, if the noisy responses of the unreliable cells are correlated.
They're not very correlated. I mean, the correlation is very low. So I said they have the same amount of correlation, which doesn't mean that. I didn't mean that they have a high amount of correlation. I said, I don't remember the number now, but at the end of the talk, I can pull it up. Okay. So that's our hypothesis. And to test this hypothesis, we looked at the fly learning and discrimination system, because as I said earlier, the fly learning and discrimination system is really well mapped out. We know all the connections, so it gives us an opportunity to test this idea on the system. So before
May I
I go
ask a question, Shyam?
yeah.
So what I understand here is your unreliable cells, they are more stochastic. So
Okay.
does it mean that if you repeat it more, or if you have more samples, does that possibly change that overlap? Because right now I'm kind of confused because it's very stochastic. So then the interpretation is that you have less overlap because you have more variations on your data. But then if you repeat it more and if you have less variations, does that make it change, you think, or no? In terms of overlap?
It shouldn't change. Well, okay, it's a good question. Let me break. Okay, there are two ways I can answer it. So here we have, for the fly data set, or let's say the mass data set, 8 trials. And we can detect the reliability, the level at which, atomicity at which we can detect it is 1/8 or 1/6. The actual true reliability could be between 3/6 and 4/6. If you had 20 trials, we'd get closer to actual ground truths, let's say. So the more trials you do, you get closer to the actual reliability and closer to the actual overlap. But so to answer your question, if you have more trials, the overlap will change in the sense that if the overlap measure now is 0.2, let's say, the actual overlap might be 0.15, and you might get closer to 0.15 more trials you do, but it's not going to change it from 0.2 to 0.8. It's not going to increase the overlap.
Okay, thank you.
Actually, if it helps, think about overlap like this. If I just pick a random trial for order A and order B, what's the likelihood that the cell will respond to both? If you think about it like that, then maybe it's easier to understand that more trials you do, it doesn't change that probability.
Okay.
Okay. And we can talk about it more too, if that's not clear. So, okay. Coming back to the fly olfactory system, it has 3 neuronal components. The first component are Kenyan cells that carry odor information. The second component are dopaminergic neurons that carry information about whether stimuli is aversive, whether it's a reward or punishment. And the third component are output neurons, or they're also called mushroom body output neurons, and they signal whether the animal is going to approach or avoid. And as I said earlier, the animal approaches or avoids depending on which set of neurons is firing at a higher rate. So there are about, so here in gray, this body over here shows two compartments. There are about actually 15 compartments in the mushroom body lobes, but all compartments fall into two categories. So the first category you have, what you have are Kenyan cells. They make synapses with the approach output neurons, and they get input from aversive dopaminergic neurons. In the second compartment, you have Kenyan cells making synapses with avoidance output neurons, and they get dopaminergic input that signal reward. And the way the system of machinery works through olfactory conditioning is as follows. So let's say you're going to train the animal with reward. So you simultaneously expose the animal to reward and an odor. So over here, you have odor that is signaled by the fourth cell. And since you gave it a reward, the active dopaminergic neuron is going to fire, and it innervates the compartment which connects to the avoidance output neuron. The way plasticity works here is when there's simultaneous exposure to odor and the valence stimulus, the corresponding synapse is depressed. So let's say you do 10 more trials. Usually they do 10 to 12 trials. You do 12 trials, this synapse is highly depressed. So later, when you just give the animal the odor alone without the reward stimulus, what happens is that this synapse strength is reduced. So the output neuron for avoidance fires at a lower rate, which means that the animal approaches. And this is the way the olfactory Pavlovian conditioning works with the mushroom body. So we captured the working of this system using three equations in our model. So the first equation tells you what I just described. The way the synapse, it decreases in strength with conditioning depending on learning parameter delta and the firing rate of the neuron. So the higher the firing rate, the more it gets depressed. And the learning parameter delta here, we don't know. That hasn't been measured yet, but for the sake of this presentation, the actual exact value is not important. And the zero indicates that you can't have the synapse go negative. So there's saturation amongst the synapses. So I'll give you a small illustration of how this works. So let's say you have two orders A and B, and you condition order A to reward and order B to for punishment. The way it works is and here I'm giving you how it works for one particular cell X. So the synapse for X connects, when you expose order A, the synapse for X connects to the avoid output neuron, and the synapse for Y, the synapse for X for order B connects to the approach output neuron. And again, let's say the response of order X to A, it's a reliable cell, it responds in 4 out of 6 trials. B, it responds in 2 out of 3 trials. And you carry out the olfactory conditioning for 6 trials. At the end of it, A is much more reduced than B because it has experienced more trials and has experienced a higher firing rate. So the calculation for A alone, the ability for cell X to distinguish order A from B would be the probability that the cell will respond to order A and the difference in the synaptic connection strengths. And so that would be 4/3. And the third equation is, if you want to calculate discrimination for the whole population, you just sum up discrimination for the individual cells. There are 124 cells, so you just sum up the 124 cells. So in our model, we used, yes.
So here we are thinking about discrimination between odors that elicit opposite behavior.
Yes,
But what about odors that have the same behavior, but are still distinct contextually?
odors that have the same behavior but are still distinct, but
Like
okay.
different food sources.
sure. So most of the discrimination experiments are done where you train. If you give. The problem with that is that if you train you're basically saying both orders have the same outcome. So it's then difficult to use that as a discrimination test. Okay, let me put it the other way. What you want to see is if the animal can distinguish A from B. So you can either have it do one task for A and one task for B, and then give A or B and see which task it does. Or you can just have it do one task for A and nothing for B. And if it does the task for A again, then you know that it can distinguish A from B. But if you give B and it does the same task as A, then you know that it's confused.
Yeah, I guess I'm thinking more of natural conditions and not like experimentally training.
Yeah.
Like being able to perceive two different odors as different, even though they're both say positive odors. It would this kind of stochastic.
Yeah, okay.
Stochasticity help with that also.
In this context, in this particular example, I have them doing different things. And most experiments, I know that they have them doing different things, but I guess then it would go down to the natural condition. You'd still have to have different tasks. They don't have to be opposite tasks, but they have to be different tasks. And I'm not a behavioralist, but I'm sure you can. There's probably some task that positive task you can come up with, two positive tasks you can come up with. And then you can carry out the experiment. And okay.
Also, are these odors or odorants? Are they a single molecule?
So
Or are they a bunch of molecules together that are making sense?
So, great question. So it turns out that both, I think, the fly and the mouse literature, traditionally, just by happenstance, they found these chemicals that the fly responded to, the mouse responded to, and they stuck with them. And most of them are single monomolecular odorants and odors. But they've also, there are a bunch of experiments, but they've also used natural odors like banana and then in orange or lime, and those are like 30 or 40 chemicals. But this, for this particular task and this particular numerical experiment model, it doesn't matter whether it's monomolecular or it's a complex mixture. I don't know if I answered your question, but
I'll think about it a little
Okay.
bit more. Like,
so okay. So there are so we tested the model with two conditions. So first condition is what we call normal training, which is the normal training that we use implies they train them for about 12 trials, simultaneous exposure, valence, and odor. And in this, you can imagine that unreliable cells would not play a big role because they don't respond. They probably respond in one or two of the trials, and their responses are also at a lower rate, so they won't have much of an impact. But if you extend the training to 100 trials, then even unreliable cells respond in about 10 to 15 of the trials, and they've responded enough that they will have an impact, as much of an impact as reliable cells. And the other great thing over here is that we did to do odor comparisons. We just used the data set that we already had of fly responses to 6 odors, 6 trials. So here's the result of our modeling experiment. Let me first explain the plot, and then I will explain the model. So N on the x-axis here stands for normal amount of training, and E stands for extended amount of training. So 10 trials versus 100 trials. We also broke the odors up into dissimilar odor pairs and similar odor pairs. So dissimilar odor pairs were odor pairs that had a correlation less than 0.2, and similar ordered pairs had a correlation greater than 0.5. And we also broke the results up in terms of unreliable cells and reliable cells. So you can see the contribution of each set of cells. Okay, so now coming to the result, on the x-axis, you have the kind of training that happened, normal and or extended. And on the y-axis, you have for each ordered pair. So I think there are about 20 ordered pairs here. So for each ordered pair, you have the useful discrimination or discrimination quotient for all the cells in the ordered pair. And okay, there are two features here. If you look at unreliable cells, the normal amount of training, their contribution to discrimination is pretty low. As I said, they don't respond enough with normal training to have an impact. But with extended training, they've responded enough number of trials. And so now, and since actually, that's not important. With extended training, with 100 trials, they responded in 10 to 20 trials, they have had an impact. And so they also contribute to discrimination now. As you can see, it increases from about 1 to like 4.8 or something. This is some normalized unit. Now, the second feature is if you look at reliable cells are quite likely to be different for dissimilar ordered pairs. So right off the bat, with normal training, they have a high contribution. But we also have the synapses cannot go beyond 0. So there is saturation of synapses. So if you take it out to 100 trials, it doesn't have any more of an impact because they've already maxed out with normal training. Now, that was the result for dissimilar order pairs. If you look at similar order pairs, again, for unreliable cells, with normal amount of training, their contribution is low. But with extended training, the unreliable cells are likely to be decorrelated for similar order pairs too. So with extended training, their contribution increases. And similarly, with extended training, reliable cell, their contribution doesn't change. And in this case, with similar order pairs, we already know that similar order pairs are likely to fire or respond similarly. So their contribution is pretty low right off the bat. So this is our model for how discrimination can improve with extended learning by making use of unreliable cells.
I have another question about the model. Thinking about, so in the cartoon that you showed earlier, it seems like this only
This one?
works. Yes,
Okay.
it seems like it only works if X is reliable for one order and unreliable for the other, right?
Yes.
But for a similar order where X might be reliable for both orders, how would it work?
Good point. So if let's say for B2 for A response in 4 trials, in B for B response for 4 trials again. But let's say instead of the 3rd trial, it responds in the 2nd trial. I'm just asking you to imagine. Then it doesn't matter, but at the end, like if you look at the depression in the synapses, it would be the same. So the synapse, it would be when you subtract the synapses, it would be a wash. So if it's reliable for both, then the order is not going to contribute, or the cell is not going to contribute much to this. And actually, the rightmost plot over here, that's what it shows, because with normal amount of training, the discrimination contribution of reliable cells is pretty low.
Oh, okay, I see the point. So an unreliable cell would not fire for both B and B.
Yes,
And
more,
so that would, okay.
yeah.
Okay, I get the picture now.
Okay.
A light bulb went off.
But I got confused. Unreliable cell is one that doesn't respond to B, so.
Okay, if you take orders A and B and you take a cell X,
Okay,
if it's unreliable for A and unreliable for B, most likely it won't respond at the same time.
okay.
Or it won't respond similarly. if it's reliable for A, but unreliable for B. With, actually, I take it back.
Okay, maybe it's another way to put it is, I don't understand how, when you have. In my intuition is that if you have a reliable cell, it should have a great contribution when the orders are similar, because it has a high confidence.
No, so the thing is, if a cell is reliable for A and it's reliable for B,
Ah, they have, okay, right.
then they cancel each other out. The synapses cancel each other out. The maximum contribution is if a cell responds for A, but it doesn't respond to B,
Yes.
then
reliable
you
for
get
A.
the full contribution. And the thing with, okay, now I got a little confused, but now the answer to your unreliable question is if you just take, if you have a population of 100 cells, you have 5 cells that are unreliable in each, they're unlikely to respond the same. Most likely what will happen is if the cell responds to A, it won't respond to B. Because it's unreliable.
Yeah.
That's what I'm getting.
So what you really want ideally, right, to optimally discriminate these odors is you want cells that reliably respond to one, but not to the other.
Yeah.
Right, so that's
Yeah, that's good. Yeah, if you can manufacture that, then it's,
right.
you're great.
So
And actually, that's what happens with dissimilar orders.
right, so what I'm trying to understand here is, so obviously, you have a learning rule that works and it's motivated by the data and that's good. But I'm trying to understand, is there really sort of a specific computational benefit to having stochasticity in the system? Or is it just that, you have this large pool of extra cells that don't do anything useful in the beginning and that respond only weakly, and your learning rule with extended training can recruit them to do something useful?
Yes.
So could I do the same thing with just a large pool of neurons that sort of respond very weakly but reliably? Again, could I recruit them to increase my similarity
Yes, okay.
or decrease?
So the second thing that you said, that's the advantage. Actually, I see that as an advantage, except if your weakly responding cells are also reliable, they'll respond the same way to both odors. So what you want is you want a second set of cells that don't respond the same way to both odors. So if they're reliable but they don't respond the same way, then you have achieved what you want.
Right. So if I have some graded response, right, I can still increase the distance, right?
Yeah, all you need to do is increase the distance.
Yeah.
So if you can manage some way where they respond reliably, but the set that responds reliably to this order is not the same as that response to that order, then yeah, you've achieved what you want.
Right. So I'm asking,
But
couldn't we do something like that with a similar learning rule without invoking stochasticity?
you could, you very well could, but it doesn't seem to be happening here. Based on the data set, that's not what's happening.
Okay.
But let's say you wanted to do a machine learning algorithm to implement this, then what you're saying seems the easier, more optimal way to go, because you have more control over how your algorithm works. Over here, you have less control over it.
Thank you.
So I have one more question, Shyam. So when you say that the contribution increases with extended training for unreliable cells, does that mean that in terms of the overlap, does it mean that it's even less overlap for similar odor for unreliable?
Yes. Yes, exactly. That's exactly what I'm saying.
Okay.
As I showed you before, unreliable cells are decorrelated across all orders.
Right.
All ordered pairs.
And what about for the, can you explain for this, for the reliable cell? So for the extended training, it doesn't change, right?
It doesn't change because there's saturation of the synapses. So
Right,
It-
right. It makes sense.
Yeah.
It makes sense. That's my, and the last question is like, have you also combined the reliable and unreliable to see kind of like the overall how it will be changing in terms of the contribution?
Yes, I don't have a graph in this presentation, but we do have combined graphs of all cells. I just broke it up to kind of show you the contributions of both sets of cells.
And do you remember like with the combined, how do they like combined population response to the similar order?
Since a combined population, so if you look at this, the increase would be about this, or the average increase here. So it would go from, let's say, 1 to 4.
Okay, so is it like more like the combined population? Is it like more like the reliable cell as a group, or is it like unreliable?
The combined population will be more like the unreliable cells for similar orders.
Makes sense, makes sense. Perfect. Thank you. That's what I was looking for.
Okay. By the way, I'm not claiming here that, I mean, I'm just claiming that discrimination will improve. I'm not claiming that you would be able to achieve discrimination, because the ultimate mechanism, if one cell is enough, if 15 cells are enough to give you a difference between the two orders, it's still a matter of experimental discovery. Okay, maybe just to get to individual, like what I showed you earlier were I combined all cells into either unreliable or reliable cells. So here we also broke down contributions per reliability subclass. So on the x-axis, we have the 6 reliability subclasses for flies. And on the y-axis, we have the amount of improvement you see with extended training. So y-axis is contributions extended versus extended minus normal training. As you can see, the highest contributions come from the lowest reliability subclasses. And the line at the bottom, this dotted line, is the zero line. And we did the same thing. I didn't show you the figure for the mouse, but it's pretty similar. And if you look at the contributions per subclass for mice, it follows the same trend. As reliability increases on the y-axis, the contributions towards discrimination with extended training increases. So our basic model of discrimination in the fly is that initially you have 4 sets of cells. If you take 2 orders, you have 4 sets of cells, unreliable and reliable for both. For similar orders, the reliable cells are the same for both orders. They differ in their unreliable cells. Initially, the connections from all of them to approach and avoid neurons are the same. So it's all washed. So the animal, if you give it one order, it just stays where it is. With a little bit of training, the connections from the reliable cells are weakened to both approach and avoid neurons. It's still a wash because they just cancel each other out. But now with extended training, that depresses the connections of synapses from the unreliable cells too. And since the unreliable cells are not common to the 2 order pairs, you can now distinguish, or you can do better discrimination.
I feel like there's an implicit like homunculus in this that's doing the discrimination. You have these like 2 populations.
Yes, where's the homunculus?
I mean, like there has to be like a readout. Like, there's an implicit reader.
Yeah, so we, all right. So in terms of the reader, we are saying that, you know, you have the approach and avoid neurons. So, I mean, the approach and avoid neurons are reading out what the synaptic inputs are. And the mechanism there, we are assuming that it's going to do approach minus avoid or some modulus of that. But it could be approach divided by avoid, it could be any number of things. We don't know what the machinery is. We just assume the simplest machinery, which was we did the modulus difference. So there's a homunculus part in we assume the simplest
Simply so much.
way to compare the two neurons, but
Like, so, like, yeah. But it's just, so where is this discrimination have, because you have your neurons that are making this discrimination have to be then innervating the entirety of the population, right? Or be innervated by the entirety of the population.
yes, so I originally mentioned that there are 15 compartments. So each compartment differs in size. The biggest compartment, so the mushroom body as a whole has 2,000 neurons. And each compartment, the biggest compartment has about 600 Kenyan cells, or it gets connections from 600 Kenyan cells. And the smallest one, I think, is about 150 to 200. And the output neurons get connections from each. Let's say one compartment has 600 Kenyan cells. The output neuron gets inputs from each of the from all the 600. But
You're not gonna expect the same thing in mouse, right?
so, good question. The mouse, it's not clear how discrimination is happening. This machinery is not known in the mouse. There are some hints. Looks like the prefrontal cortex might play the same role as like output approach neurons, and the amygdala does aversive versus rewarding stimuli, according to that, but it's nothing close to as simple as mouse machinery. So it's not clear where the mouse discrimination is happening, but we do know that in the mouse, you need the peripheral cortex cells to discriminate. And it so happens that the mouse, up to the peripheral cortex, there's a large conservation of architecture, so the responses are similar. So downstream of that, it's just an abstraction. We assume that there's some machinery which does something similar to the fly, but as just an assumption, and most likely it's going to be much more complicated than anything you think about. Okay. So that's our model for discrimination. And I think I've gone over time, so I will quickly tell you the third part, and then I'll stop. If you want more details on the third part, I'm happy to talk about it at length. I can hang out and we can talk more. So the third part, we wanted to know what generates the stochastic code that we just showed. And how much time do I have? 2 minutes?
I don't know. We're done when everybody walks out.
Okay, all right. So, okay. I'll be kind to the audience and I'll finish in 2 minutes. So basically the idea is here, we again use the flyover factory circuit, and we use the flyover factory circuit because this is a circuit which is pretty well known, the architecture is known, and we also know the connection strengths of these two parts of the circuit. So we can mimic the circuit in a model without using too many free parameters. So you have glomeruli connecting to Kenyan cells, and the Kenyan cells are the winner-take-all circuit that suppresses all of the highest firing neurons. And like I said, I'll go through this quickly. There are previous publications over here, Bandavik, Sophie Caron, Gruntman, Stevens. They all calculated the connection structure of different connection structure of the circuit. This circuit. So we developed a linear rate firing model. So the order, color showed the intensity of firing or the intensity of the connection strengths. So for the order, we chose the same intensity, the same distribution as was found in Vikas Bandavik's paper. And for the connection strength, we chose connection matrix from projection neurons to Kenyan cells. We chose the same connection matrix that was published in Sophie Caron and all. And there have been subsequent publications, the Fly EM data set, which shows what the connections are between the Kenyan cells and anterior paired lateral. So we use the same connections. So basically the point is, we had this model, we developed this model, and we introduced noise in various components of the model. So there are 3 neuronal structures: glomeruli projection neurons, Kenyan cells, and the APL neuron. And you have 3 connection structures between them. And we introduced noise, multiplicative noise also Gaussian multiplicative noise. And we saw which ones give us the results that we observed with the actual data. So those results we wanted to observe were, for example, over here, on the x-axis, you have noise. On the y-axis, you have the ratio of reliable to unreliable cells per trial. And the dotted line shows the actual experimental result. So as you vary the noise, you see which one gets you closest to the experimental result. And I'm just showing you two examples here. In the interest of speeding up, we manipulated a lot of the parameters simultaneously, 6 parameters, and we varied them from 0 to about 50% or 40%, because there's been published data that shows that up to 40%, there's been up to 40% observed noise in projection noise. So we use that as a marker. And I'll just show you the main result. My battery is running low, so it might shut off any point. So before that happens, what we saw, the most important parameters were noise in Kenyan cells, projection neurons to Kenyan cell connections, and APL to Kenyan cell connections and APL. And we found that over here, we had about,
I think it's charging.
okay, charging your thing.
Yeah, potentially.
So what we found is that we had this multi-parameter, 6-parameter set, and we had about 46,000 points in this parameter space. There are also two other parameters, 44,000 points. Sorry, everyone. I ran out of power. So the whole time I was just charging it.
No.
Okay, let me just finish up quickly. So we looked at all the parameter sets and we saw that for every successful parameter set, so we looked at reliable to unreliable cells, and we looked at also, as I showed you, the response rate, the reliability curve, and also the reliability versus number of cells in each reliability class. So these 4 results, we saw which sets gave us all 4 results at the same time. And we found that all sets that gave us successful results required the APL and or noise in APL or APL to KC feedback connection. So our candidate for producing the noise is the negative feedback loop from APL back to the Kenyan cells. And okay, that's not good.
So that does that mean that the same neurons are unreliable for all, because there's one APL neuron that has strong connections to some Kenyan cells and weak connections to other Kenyan cells?
So I think on the whole, the APL connection strength to most Kenyan connection feedback inhibition strength to most Kenyan cells is about the same. But there are multiple synapses, so if you average them out, if they're picked from some distribution, but there are multiple synapses, if you average them out, they're probably going to come up within some range. And the way, so let's say, actually, I haven't shown here, but we found that the Kenyan cell population without APL, if you silence APL, it has a gamma distribution, or the response rate follows a gamma distribution. So, and if you take a lot of cells, maybe it's like a Gaussian. So let's say you have about 85. Let's say APL subtracts everything but the top 85, beyond the top 50% of cells. If you have noise in the feedback connection, then if the noise is Gaussian, it's, let's say you're tossing a coin, all the cells that fire close to the 85% are going to be unreliable cells, because you're tossing a coin and it's either silenced or not silenced. Cells that are closer to like the 95th percentile, you need a lot of noise to to silence them. So they are your reliable cells. And the ones along the border are more likely to be like 50% reliable. And the ones as you get like towards 75th percentile, those are more likely to be our most unreliable cells. And beyond that, they're most likely to be mostly silent cells. So that's the general mechanism that we propose as a candidate mechanism. And we are doing further experiments now to figure out whether this is actually happening in the fly. And in the mouse, there are more complex feedback circuits. So we're trying to figure out what those circuits, if those circuits might also be contributing to stochasticity. Okay, sorry for going over time, but we also started late. Thanks for coming. I like all your questions. And thank you for listening to.
I have one more
Yes.
question. So, if you, I don't know if you can do it, but if you try to categorize the odors that were used in the experiment to, innately aversive, innately appetitive, and that's in neutral. Is there a difference in the fraction of unreliable cells for each one of these odor categories?
Okay, I'll answer your question in two parts. So the first part is there's a publication by Gero Wiesenbach, I think 2017. I don't know if you probably referring to that.
No.
He categorized a lot of odors that people use for flies, and he found that most odors have some innate
This
awareness to them.
is this trap assay.
I forgot what. method
Yeah,
it was.
well, okay, yeah, we can discuss more. Our collaborator has some, I think, reasonable criticism of the experiment.
Okay.
Yeah, okay.
So what I know from that paper is that most odors have some innate aversion or attractive valence properties. So if you take two such odors and you do training, I think you would have to do more trials than that. If you take an innately aversive odor and you want to make it a rewarding odor, you'd have to go through more trials to get it rewarding. And it's a great question. I don't know if, so in this, one of the assumptions is that the odors are neutral and then extended training has a benefit. So I don't know the amount of benefit extended training, how much extended training you can do with that and how much benefit. Like I imagine that convert an innately aversive odor to a rewarding odor, you might not be able to go to the full extent of extended training. But the second question you asked, or main question you asked, is that unreliable and reliable cells different? No, my answer is speculations that they won't be different, or the statistics of the unreliable to reliable cell population won't be different.
But could we test that using this this experiment that you mentioned? And Scott, what is
Yeah, actually, I think because we can go back and look at the orders that were used in the order set, and then you're saying that paper is controversial.
I mean, it's not super. They're just. There's some point in it that might bias the results in one way.
Okay. So we can go and look at that data set and then look at this data set and compare, see like if innate aversion is like 50% or 60%, and if the statistics changes with that. So that calculation can be done. It did strike me, but
Okay,
thank you. All right.
Any other questions online? In person? You're welcome to join us for tea if you're interested.
Yeah, fine.
Oh, there's some chat questions.
Oh, there are
No,
challenges.
it's Sarah. Just between, yeah.
Okay. Let's see if I don't. Hey, I missed this composing for the chat.